Okay, hi everyone. I am going to continue this week talking a little bit more about humans and microbes, their relationship with us, and also some of the features of the pathogens. Last week, the video was talking about, if you saw the video given on the schedule, was just an introduction, a very basic introduction to pathogens. And we know one thing is that pathogens cause damage to the host. So, again, I'm going to just review a little bit about humans and microbes and our relationship with them and talk about those that do cause damage to us. So, first of all, as a review of host and microbes, of microbes and us. And we know that there's a symbiosis. That relationship is a symbiosis. And there are uh, three, three phases of symbiosis. The first two interactions can be mostly beneficial to the host. That will be mutualism. So in that case, we've got the human host benefits and the microbe benefits. An example of that would be Escherichia coli. We know that they live in our gut, and the Escherichia coli uses the food that we eat for nutrition. They can use that food for their nutrition, but as a result, one of the metabolites that they produce is vitamin K, which we need. In commensalism, this also is kind of beneficial. Not, it doesn't affect us, but it does affect the bacteria beneficially. And an example of that would be your Staphylococcus epidermidis. Now we know that they live on our skin, and that what they do is they use our dead skin cells to provide nutrients for them. What do they give us in return? Nothing, but they do not harm us. There's no effect on us. Finally, and this is what I'm going to be talking about in this unit, is the interaction in which the microbe is harmful to us or to the host. And the term for that is parasitism. So every pathogen undertakes parasitism. Okay, so an example of this would be Bacillus anthracis, now which causes anthrax. That can be found in animals and also humans, and it can be deadly, especially years ago. Before they can treat it now to some extent with antibiotics and antitoxins, but it is typically a very harmful bacteria, and it is of the Bacillus genus. In fact, one of the only really pathogenic bacteria from the genus Bacillus. They normally do not cause that much trouble for us. So again, they do benefit us, and this again is just a overview of the terms microbiota and microbiome, because I'll be referring to that again in the next couple of slides. So anyhow, we know that the microbiota is a collection of those organisms or those symbionts, those that live in symbiosis with us. And they are found in all plants and animals, okay? Now, an interesting thing, too, is that 90% of the cells that we have are microbiota. So 90% of our cells in and on us are microbes, okay? And the gut microbe holds the most of them. Approximately, it weighs, if you would take all those microbes and weigh them, they weigh about the same as the human brain. And of course, you know that includes bacteria, archaea, also your protozoa and fungi and viruses. So when I talk about microbiota, I am not just talking about bacteria. But of course, that's what we concentrate on because it is microbiology, and we do concentrate on bacteria in this unit. And anyhow, we have found that our microbiota is overall crucial, very important and crucial for our immune system, for the production of hormones and also the metabolites they make keep us in homeostasis, okay? Homeostasis. Of course, some of those microbes are also those parasites, okay? Parasitism. And those are the pathogens. That I'll be talking about later in this slideshow. The difference between microbiota and microbiome is that, of course, a microbiome, if you think of genome, are the genomes, all the genes of the microorganisms that reside in a certain niche. So we would call the microbes, the genes of the microbes that live in our gut, the microbiome. So anyhow, 99% of the genes needed for homeostasis reside in our microbiome. Now that you look at that, you say, that doesn't sound right. What's going on here? You mean that we don't maintain, or our cells don't maintain homeostasis? No, not in and all of themselves. In fact, many of them, the products of the genes of the microbes, when expressed, keep our cells in homeostasis. So here we go. I've got two rows, row A and row B. And row A is all those different colors. Each color represents a certain phyla of bacteria. And they were taken from 6 sites on the human body. That first frame with all the red was taken from our nostril. Then we have the second frame behind the ear. We've got samples taken from inside the cheek, the mouth, the top of the tongue, and finally the stool. Now that second row below it, B, are metabolic pathways. So each one of those little frames, those 6 frames, are the metabolic pathways that occurred at each site. So for example, if you look at the A, the first frame is mostly red, and that is made up mostly actinobacteria. That was taken from the nostril. You've got a little yellow, which is proteobacteria. If you look at the first frame in the row beneath it, metabolic pathways, you'll see that there's a whole assortment of colors: pink, reds, blue, greens, yellows. And that each color there represents a certain metabolic pathway. Now, if you notice, going across the row for B, you've got basically very similar colors, a very similar pattern in each frame. Whereas in the phyla in row A, you've got different colors where one may be predominantly red. The second to last frame is predominantly blue. So it is showing you that even though at each source there may be different phyla, they actually will carry on the same metabolic processes. It doesn't depend on where that is, okay? And what are some of those metabolic pathways? Well, we've got basically the breakdown of sugars. We've got ATP synthesis. We have pyrimidine metabolism. So what it's saying is at each site, we've got bacteria, no matter what type or phyla they are, that carry on the same specific pathways. Okay? And this is what keeps us in homeostasis, our cells in homeostasis, those products or byproducts of those metabolic pathways will keep our cells in homeostasis. Now, how do we acquire this bacteria or these microbiomes? There is a good little tape right there, video, how our microbes make us who we are. And, view it if you'd like. It's a TED Talk. It's not very long, but it's rather interesting. But basically, he is saying what is right here in the slide. How do we acquire our microbiomes? And I'm going to focus here, we're focusing on the gut microbiome because that's a very important microbiome. And anyhow, a lot of it depends upon as early as birth and delivery. So if it was a vaginal birth or a cesarean, you will acquire certain bacteria depending upon the mode of delivery at birth. Once you've acquired the initial bacteria, then we've got early colonizers. So this is between infancy and 2 years old. And you acquire this bacteria usually through your diet, the gut bacteria especially through your diet. And some of these would be your firmicutes, your staphylococcus, streptococcus, some gram-negatives of the Enterobacteriaceae. And again, the diet here would depend on whether you were breastfed or formula fed. And then when you start eating solid foods, you will pick up some more species of bacteria. So that by the time the infant is 2 years old, they have reached a pretty stable microbiome. Okay. Now, by stable, I mean you have your general basic phyla that have been established in your gut and also, within other parts of the body. What happens when you're from 2 years old on to adulthood is that it may fluctuate. It may even be, it's different from one individual to another. And a lot of it depends upon your diet. If you eat a lot of protein or animal fat, you will acquire a certain phyla or phylas. If you eat a lot of carbs, that again will introduce different phyla of bacteria. So then, if you've got a healthy microbiome, a gut microbiome, your gut will be in eubiosis. So what does that mean? It means that you've got a microbiome that basically is sharing a group of functions, a specific type of metabolic pathways, and it will be a state of health. Now, at some point, you may undergo, or a person may become a microbial dysbiosis, where the gut is in unbalance. And of course, that again, it depends upon diet. You may not be eating enough. You may be eating too much or too much of one type of food. So a certain type of food that's bad for you will stimulate the production of bacteria that aren't necessarily good for you, okay? And that they aren't producing what your body needs for homeostasis. Another thing, they've also found that diet may also lead to, a bad diet or a poor diet lead to allergies, asthma, type 1 diabetes, irritable bowel disease, and again, multiple sclerosis. Now, what is the relationship between multiple sclerosis and dysbiosis in the gut? They aren't really sure on that. Then again, you may get infections, okay? You may get a disease of the gut. They treat it with antibiotics, too many antibiotics over a long period of time, and you will have an overtake or takeover by opportunistic pathogens, which are pathogens that may be living in the gut, but they have not really have not caused any damage because the good bacteria a lot of the good bacteria is living there. But once you've had antibiotics, a good bacteria, of course, we know may die off, and you will have bacteria such as Clostridium difficile, which causes infection and disease, take over. So anyhow, another function also, or another thing they have found out is that there is a connection between the gut microbiome and our brains. Okay? And what happens is you've got the gut microbiome producing things or producing metabolites such as short chain fatty acids. It also may be producing proteins that will keep the lining of the intestine nice and tight junctions. The metabolites will also be producing products that will signal immune system and keep our immune system kind of toned up. And then also you may have neurotransmitters such as serotonin. A large percentage of serotonin is produced in the gut by the microbes. So what happens is these neurotransmitters can be transmitted to the brain, and they actually will control our behavior or affect our mood. So you've got serotonin produced. It will actually, by what we eat, by what's produced by the microbes in our gut, have an effect on our mood, make us feel good. There are feel-good foods that when eaten, digested by the bacteria, will affect our mood. Okay? And anyhow, there is a connection. So I said a connection between the brain and the gut. Through the vagus nerve, these transmitters will affect our mood or behavior. And then in return, the brain will affect or can affect our gut. So for instance, if you are lacking in a certain neurotransmitter and you become anxious, okay, that anxiety or stress response will be produced by neurotransmitters in the brain, and it can have an effect on your gut by causing peristalsis. You may get too much constriction and have diarrhea, or you may have lack of peristalsis and become constipated. So it's kind of a cycle. The gut microbiome not only keeps us in homeostasis, but also affects our brain. So this is getting a little closer look. We're honing in now on the lining of the intestine. And in eubiosis, when you've got healthy bacteria present or non-pathogenic bacteria present, you have little inflammation. You have a decrease in oxidative stress, which means you have a decrease in the production of free radicals. Of course, your gut microbiota is in balance. You've got, like I said, good bacteria, plenty of them, a diversified group of them that either are beneficial or commensals. There also is a very protective mucus-like layer on top of covering the cells lining the intestine. And you have intact tight junctions. So intact tight junctions, that tight junctions are what hold the cells lining the intestine close together. So there is very little chance of bacteria or large proteins passing through to the blood. At the metabolic level, in eubiosis, you've got increase in the production of antioxidants, increase in indole and butyrate. So these are important, important metabolites for us. And you basically have a decrease in signaling factors that cause inflammation. Finally, the cellular response in eubiosis, again, you have apoptosis, which means cell death, and a decrease in cell proliferation. So what that means is you do have cells dying at a healthy rate, and you have a decrease in cell proliferation, which means they aren't over-dividing. So in other words, you look at that eubiosis, life is good, everything's balanced. Now, if it's dysbiosis, so what would cause that? It would be, it can be diet. It can be, in this case, it may be a pathogen, an active pathogen who has come along and is multiplying. It basically has either killed off or the good bacteria has died off, many of it, because the pathogen is taking over the intestine. And what happens is you also may have some pathogens, they're opportunistic pathogens, so they are there, like C. diff, in a small amount, but normally they don't cause any problems. But now that you've got dysbiosis and a decrease in healthy bacteria and an increase in the pathogens, these guys, these opportunistic pathogens, can now multiply. So some of the things that go on with dysbiosis is you have an increase in inflammation. Okay, the body realizes that something's not going right. You've got more free radicals being produced. The mucin layer that covers the intestinal cells has all but broken down. And as a result, you've got inflamed cells of the intestine, the enterocytes, and you lose that tight junction. So the bacteria now, some of the opportunistic bacteria or even the pathogens, may seep through those tight junctions, and this leads to leaky gut. At the metabolic and molecular response, we have a lot of inflammation going on and a lot of signaling cytokines that basically add to inflammation and also to cells, immune cells response. At the cellular response in dysbiosis, we have a decrease in cell death, an increase in cell proliferation, among other things. So what is happening is you've got the cells are not dying. In fact, what they are doing is super dividing. This happens in dysbiosis after a long term of inflammation. And this cell proliferation may lead to overgrowth of cells and even to cancer. So over the long run, dysbiosis can cause some hefty problems to us as humans. There have been studies about the gut microbiome, and they have done some studies that they are able to see that people that have multiple sclerosis have certain characteristic microbiome, and they're trying to see what effect that has on the disease, or does the disease affect the microbiome in the gut. Also, they've tested people with fibromyalgia and have been also trying to determine the composition of their gut microbiome. I mean, maybe at some point they'll be able to give oral or fecal transplants to patients with these diseases and see if that helps with the disease conditions. And in fact, I know some of you have probably heard about the fecal transplant in people with C. diff, where they've taken a fecal matter from a healthy individual and have transplanted that into uh, the intestine of someone that is um, that has C. diff, and it has been very promising because many times this has cured that patient after they've had this transplant, has cured them of the C. diff infection. Well, they've also done some transfer therapy, fecal transfer therapy in children, individual children with autism. And usually this autism is defined as a disability in which the individual shows certain signs and symptoms, among them the lack of communication or the repetition of behaviors. And what they've done is they've transplanted fecal matter from healthy individuals into children that are on the autism spectrum. And it seems to be very promising. If you see here in the slide, at the start of the study, 83 of the participants were rated as severe. They had severe autism. At the end of the study, only 17% were severe, 39 were mild to moderate, and 44% were below the cutoff for mild autism spectrum disorder. So this could be very, very promising. My daughter suffers from autism. She's very high on the spectrum. But it's just fascinating that how much they are linking now to our microbiome and certain diseases, not necessarily infectious diseases, but just certain diseases such as autism and multiple sclerosis. So I'm going to end here right now, and we'll continue tomorrow. I'll probably try to make it up tomorrow or Wednesday for sure, talking more about infection and disease and some of the features of those pathogenic bacteria that make them so dangerous or damaging to us. Okay? Hope you all have a good night, and I will see you soon. Bye.